I want to thank Adisio for giving me this opportunity to come here and interact with experts in the field. And one good thing about being one of the last speakers is the speakers before me have done most of the job. So I probably have to just go very quickly. I was joking with somebody in the afternoon that I should just pack my bag and go to the airport because they've spoken for me. But there are a few things which I want to emphasize, which is some of the work we've done on the gut, but also outside the gut. Like I said, a lot of things about butyrate source has been said. We do know that butyrate is a product of fiber fermentation, but it's also can be produced endogenously. In fact, it's one of the things that contributes to the smell of body odor. So if you see somebody that has a very strong body odor, maybe they actually have a lot of butyrate, so it might not be a bad thing after all, considering all the good things we've said about butyrate today. It's also been said that butyrate can be produced by several bacterial groups that have the various enzymes, mostly from acetyl-CoA. That has been discussed very well today already. But there's a lot of interest about butyrate being applied not only in nutrition research, but in neuroscience research as well. I'm going to be presenting some data on that. As far as animal nutrition is concerned, butyrate is being supplied as sodium butyrate, but also as tributyrene, which can be broken down into 3 butyrate units. So butyrate, we can get supplement butyrate for so many reasons, also in different forms. And butyrate is very important as far as gut health is concerned. Hippocrates said all diseases originates in the gut. And that just shows us clearly the role of the gut as an interface for health and disease, but also for performance. Okay, because the GI tract is integrated for regulation of various body functions. And if you look at all these functions, butyrate has been shown to interact with virtually all these functions. Okay, so butyrate is a very key molecule in regulating many of these functions. Okay, we know, we've talked about it today already, it's an energy source. Okay, it's implicated in obesity regulation. I'll show some data, especially in rodent models, in immune system regulation, in cancer, and of course, in brain function. And the question people always ask is, do we get enough butyrate in circulation to justify some of the attributes that we are claiming that butyrate is responsible for? And the question is, yeah, we may not get the same level of butyrate that we see in the gut, in the peripheral circulation, but there's butyrate in circulation, okay? This particular work is a human study, and what they have done is to feed different kinds of fiber to people, and then they measured the amount of butyrate, okay, in circulation. So in whether you feed barley bread or white bread, it's associated with increased level of butyrate in circulation in humans, and that's also associated with lower level of glucose and lower level of lower circulating concentration of insulin. So clearly you can pick up increased butyrate in circulation when you take fermentable fiber, even in humans, okay? So butyrate responds to diet, even in humans. So it's not just that its level is increased in the gut, but even peripheral circulation. So it must be doing something if it's in peripheral circulation, okay? And one of the things it did was to reduce the level of inflammatory cytokines such as interleukin-6, and TNF-alpha. So clearly, butyrate is associated with reduced peripheral circulating inflammatory cytokines, even in humans. Okay. But Newton, that we know is an expert on fiber, also showed that if you give rye to pigs and you measure portal and the mesenteric artery blood, you also have increased circulating butyrate concentration. So the same thing in humans, in pigs, of course, in chickens and other animals that eat a lot of fermentable fiber. Okay, but like I said, we do know that butyrate acts first in the gut, because the cells of the gut, the epithelial cells, have the first stab, okay, at the butyrate that is being produced, especially in the colon. So most of the effects of butyrate that have been characterized have been in the gut, where it serves as energy source, helping, in proliferation and maturation of the tight junctions, but also in immune response as well, in training the immune system. Okay. So I'm going to be talking briefly on some of the things that happen in the gut, in the nervous system, in peripheral tissues as well. So those are the three things I'll be talking about today. So let's look at some of the effects in the gut, and it will repeat some of the things that have been said today, but a few instances I'll be mentioning some things that have not been emphasized. And in this particular paper, I had a former PhD student use the IPEC-J2 cells. These are porcine intestinal epithelial cells to try to study some of the mechanisms of butyrate in that cell model, again, in trying to understand very detailed actional, since and this is very appropriate for this meeting because we're trying to understand the mechanism of butyrate effect. Again, some of the things have been mentioned earlier this morning, but we do know that maintaining a proper tight junction is very important for animals. Again, when you compromise that tight junction, you break up you mess up the integrity, then you allow a lot of antigens or microorganisms and toxins to pass through into the body. And so maintaining those tight junctions is very, very important. And that was why we focus on that in this particular study. Most of the tight junctions that we focus on are the cloudins or the occludins, again, that they help to maintain that tight junction, preventing a lot of paracellular flux of toxins and things that may be dangerous for the body if they get through into circulation. So the question we asked was, can butyrate protect IPEC cells or these cells from LPS-induced damage on tight junction expression? One thing we've emphasized today at this meeting is that maybe under condition of stress, okay, whether it's inflammatory stress or even temperature stress, maybe that's when you see a beneficial effect of butyrate. And that was why we use this model of stress to see if butyrate can help mitigate that. Okay, and we saw when we look at the expression of some cytokines, especially interleukin 8, we saw a very slight but significant induction of that in the presence of butyrate. You can see the level is very small, but this is not too surprising because we know that germ-free animals actually have impaired immune response. Okay, so and then when you colonize those germ-free animals with bacteria, there you have their enhanced immune response. So butyrate may be helping to train the immune system as well. Okay, not to be inflammatory, but to make it very effective. Okay, and then we began to look at the intestinal paracellular uptake using the co-culture system, and we measured the uptake of this fixidextran, which is such a big molecule, it cannot be taken via the transcellular pathway. Okay, cells cannot absorb it. It can only go through the paracellular pathway, and we saw that lipopolysaccharide, again indicating or representing bacterial infection or activation of toll-like receptor, you see increased feedchannel transport, which is suppressed by butyrate. So clearly, butyrate can help suppress paracellular transport of material that is not supposed to go through. And then we began to probe the molecules, okay, the proteins, the tight junction proteins that are involved. And we saw that with bacterial lipopolysaccharide, you have suppression of tight junction proteins like claudin 3 and claudin 4. But butyrate, especially at the 1 millimolar concentration, reversed this suppression, so helping to preserve tight junction protein expression. But this effect was selective in that we saw it with claudin 3 and claudin 4, but not with ZO1. So it's not a generalized, unregulated effect. This shows that butyrate is actually targeting specific pathways to regulate the expression of these genes. So again, it's butyrate, as the title of our talk today, it's a messenger, it's a signaling molecule, okay? So that this kind of results confirms that butyrate targets specific pathways, okay? And for bacteria, endotoxin, to activate inflammation, they have to bind to toll-like receptor 4 in particular for gram-negative bacteria. What we saw was that with butyrate, you have decreased expression of toll-like receptor 4. Again, that might explain some of the anti-inflammatory effect of butyrate, okay? Decreasing the expression of this receptor so that ligand-receptor interaction is reduced. And then we began to look at the mechanism for protein synthesis. Okay, if butyrate could affect this mechanism as well, we do know that our tight junction proteins, which we measured, they were increased. The question is, can butyrate also regulate protein synthesis machinery? And that's exactly what we saw. I don't have a pointer here, but clearly, activation of AKT by insulin stimulates protein synthesis, okay? That's how protein synthesis is stimulated. So that's how protein synthesis is stimulated when you activate AKT, and then this is the system that leads to stimulation of that protein synthesis initiation complex. You have this 4EBP, 4 eukaryotic binding protein. And we could see that this lipopolysaccharide suppressed the activation of that protein by phosphorylation, and butyrate reversed that. Okay, butyrate increased the phosphorylation of AKT. The other thing that butyrate did was to increase the phosphorylation of this 4EBP. Okay, again, increased phosphorylation is associated with activation of protein synthesis. So butyrate activated protein synthesis so that, and that was partly responsible for why butyrate restored the expression of claudin 4 and claudin 3. Okay, so again, more insight into the mechanism of butyrate at the cellular level. But butyrate is also a source of energy, and ATP is the energy currency of the cell, and what we saw was that butyrate also increased the intracellular concentration of butyrate in this epithelial cell. So butyrate is working via different mechanisms, increasing the energy level of the cell, down-regulating inflammation, increasing protein synthesis of tight junction proteins to ultimately enhance integrity of the tight junctions. Okay, so that is a summary of what how we think butyrate is acting, to suppress inflammatory cytokine. The other ones which I didn't show was interleukin 6 and TNF-alpha, which were clearly downregulated. But butyrate is increasing AKT signaling, helping to increase protein synthesis, helping to maintain the integrity of the tight junctions. So how about effect of butyrate in the brain? Like I said, butyrate exists in peripheral circulation. Okay, we do know that, of course, that amount is not as high as in the gut, but it is there. Okay, it is there. Even in the brain, you can find butyrate, even very small amount. And that's why a lot of labs all over the world are trying to investigate whether butyrate is implicated in this gut-brain axis, okay, the communication between the gut and the brain. Clearly, this is an area with a lot of interest. And how could butyrate be implicated, play a role in this gut-brain axis communication? Well, we know that butyrate can lead to secretion of things like PYY, like GLP-1. It was mentioned earlier this morning. Those hormones act as endocrine hormones. Okay, they act in distant places where they are than where they are produced in the brain. PYY suppresses appetite, for instance. But butyrate doesn't have to just release endocrine hormones. It can actually stimulate vagus nerve-dependent activation of the brain as well. And we do know it can regulate the immune system. There's a lot of data on that. But also it can act as a signaling molecule in the brain. In other words, it binding to its receptor in the brain or all those other peripheral tissues, it can act via that mechanism as well. So multiple mechanisms could explain why butyrate could activate or regulate that gut-brain axis communication. Okay, there's evidence that butyrate may be able to cross, this is the blood-brain barrier, okay? There's evidence that, yes, that could actually happen. And that's because butyrate induces a dose-dependent increase in glia, or neuronal or glia. These are macrophages in the brain, nuclear histone acetylation. Okay, with higher level of butyrate, we know that butyrate is a histone deacetylase inhibitor, okay? That's one of the mechanisms of butyrate. So butyrate actually, leads to higher level of acetylation in the brain, even though it's given orally, okay? So that means it's making its way into the brain to affect the level of acetylation in the brain. And as shown from studies at Karolinska Institute, it was shown clearly that germ-free mice have increased permeability of the blood-brain barrier, okay? And when you give butyrate, you restore that blood-brain barrier. So clearly, butyrate is also linked with maintenance of blood-brain barrier, integrity, okay? So clearly, butyrate can act in the brain. Okay. However, the interesting thing is that butyrate does not necessarily need to enter the brain, okay, but it can also indirectly influence processes in the brain in order to affect brain function. Okay, it doesn't have to make its way to the brain, but can even hang distally, even from the gut, leading to changes in the brain. Okay. Again, mechanisms such as release of serotonin, okay, butyrate can lead to serotonin production, and then we know what serotonin does. It can activate vagus nerve via this 5-HT3 receptor, and of course, vagus nerve we know that can affect the brain. It can also stimulate PYY and GLP-1 production from enteroendocrine cells. So it can act distally and yet affect the brain. For instance, with butyrate and acetate, you have increased expression of the gene tryptophan hydroxylase 1 that leads to serotonin production. Yeah, you can see clearly butyrate leads to increased expression of that gene. And like I mentioned, the vagus nerve can go straight to the brain to affect processes in the brain. So when butyrate stimulates that nerve, it can lead to effects distally in the brain. And like the other thing that butyrate does is the regulation of inflammation, okay? This, again, microglia, they are the macrophages in the brain, and it's shown that germ-free mice, they have an immature and less active microglia. Okay, so the cells, their immune system are not active, they're immature. And this is normalized by adding a short-chain fatty acid cocktail consisting of acetate, propionate, and butyrate to the drinking water. Okay, so butyrate helps to normalize microglia activation state or maturity. But butyrate or short-chain fatty acids, they not only inhibit microglia, but we do know that they support precise tuning to ensure necessary functioning under non-inflammatory condition. And that is what we need to understand more about butyrate, how it affects this precise tuning of the immune system so that it's well prepared, well activated. But we learned a lot of this from germ-free mice experiments. But the other thing that there's a lot of labs around the world are trying to study is this concept of inflammaging, okay, which is aging that results from inflammation, okay, because we do know that inflammation affects so many organs. And we know that butyrate, because of its anti-inflammatory effect, in brain resident macrophages, can reduce this NF-kB signaling, inducing apoptosis and neuroprotection, and helping to dampen excessive inflammation in the brain. And again, doing that helps to reverse aging or slow down the aging process. This particular paper, and I know we've talked about Tregs this morning as well, it shows that these regulatory T cells, which are they help to dampen immune activation, they express FOXP3, and with sodium butyrate, you have an increase in their expression. You have an increase in these FOXP3 expressing cells. So again, indicating that butyrate can promote can have anti-inflammatory effect by regulating this Treg differentiation. A lot of work my colleagues at University of Illinois looking at how butyrate can regulate inflammation in the brain. Most of these were done in the mouse model, but clearly what they have shown in microglia, again, these are macrophages or the hippocampus, is a specific section of the brain. If you look at the expression of interleukin-1 beta, okay, very pro-inflammatory, inflammatory gene, again with sodium butyrate in the presence of LPS, sodium butyrate leads to reduction in that inflammatory gene. But you can see that these are the aged mice that were old. They had more interleukin-1 beta, and butyrate reversed that. Also did that in adult mice, but more significantly in the old mice. Same thing in the hippocampus, reduced level of interleukin-1 beta in aged mice. So butyrate, can be a source of reversing the aging in the brain, okay, especially as expressed in this model in the mice. And that's why there's a lot of interest in that. Some potential mechanism, I did mention about AKT in the epithelial cell system. In this particular model, they use vascular occlusion, okay, to cause dementia in mice. So vascular dementia, okay, again another form of brain disease. And what you see is that brain-derived neurotropic factor is increased when you add butyrate. Okay, these are butyrate-producing bacteria now. So butyrate-producing bacteria increase the expression of that brain-derived neurotrophic factor, increase the expression of phospho-AKT, okay, which I also mentioned with epithelial cell. So clearly, butyrate can target brain or brain cells. And this bacteria, of course, Clostridium botulism, can measure the amount of butyrate in the brain. it's very small, but compared to the control, which were not given this bacteria, there was slightly increased expression of level of butyrate in the brain. So clearly, this butyrotropic bacteria, butyrogenic bacteria, are, clearly affecting the amount of butyrate in the brain, and that's having a positive effect on brain function under this vascular occlusion model. Okay, we just concluded this experiment in pigs, where we fed pigs inulin and cellulose, and at the end of a 4-week study, we measured the expression of interleukin-1 beta and superoxide dismutase in the hypothalamus of the brain. Of course, we saw a reduction in the abundance of interleukin-1 beta compared to cellulose, which is poorly fermented, but also increased expression of superoxide dismutase in inulin-fed pigs, potentially via a butyrate mediated effect. Okay, so clearly this process is also applicable in the pig model as well, perhaps in the chicken as well. Okay. But before I go to the effect of butyrate in other peripheral tissues like muscle or adipose tissue, we do know that, for instance, when you regulate the expression of serotonin or the level of serotonin, okay, this is associated with depression in humans. But in livestock, since it's a livestock conference, low level of serotonin is being associated with increased aggression in animals. Okay, so butyrate, for instance, increasing the expression, the level of serotonin, can help dampen or reduce the level of aggression in animals, and there's a lot of work in this area as well. So we talk about happy chickens. Maybe they're happy. We may not be able to tell, but we can tell that they're better if they are less aggressive, maybe by increasing their level of serotonin production by giving them butyrate or butyrogenic bacteria. But work has also been done in peripheral tissues, and the first one in mice was what was done to study effect of butyrate on insulin sensitivity and energy expenditure, and clearly it was shown that butyrate would increase the rectal temperature of the mice, but also increase the amount of brown fat, or you can see the size of the cells here. These are fat cells. The control under high-fat diet were large. With butyrate, they were smaller. So clearly, butyrate is programming the fat tissue. And we talked about butyrate being a ligand for PPARs. One of the effects of that is increased PPAR gamma co-activator that we can see in this particular Western blot. So clearly, butyrate is stimulating metabolism, okay? And the consequence of that is the reduced weight gain of the mice, okay? Some of the work I've done, I've used commercial, but also a biomedical pig model called the Osoba pigs. And for those from Spain, it's a cousin of the Iberia pigs. The Spanish brought them to the US about 300 years ago. They put them on the island of the Osoba Island. So they are very, they have metabolic syndrome under high-fat diets. And some of the work we've done, we investigated effect of inulin. And clearly what we saw that inulin reduced the level of adiposity in these pigs, but also increased expression of genes related to oxidation. Again, it agrees with findings in mice. Perhaps the same thing is applicable in humans as well. Some of the other potential impact of butyrate comes from a study we did with resistant starch. So amylose versus amylopectin corn. Amylose is highly fermentable. And what we did was to look at the quality of milk, for instance. Okay, under high amylose corn, you have increased milk fat percentage, you have increased milk total solids. Okay, so again, and pigs have very low butyrate in milk concentration, but that butyrate can actually serve as a precursor okay, for things that go in the milk, either the fatty acids or some other things, which can enrich the energy value of the milk. Clearly, a lot of work needs to be done on feeding butyrate to gestating sows. Okay, there was a question about adding butyrate in eggs. Okay, so again, butyrate is a histone deacetylase inhibitor, and that is associated with epigenetic regulation of gene expression. Okay, so maybe, again, the earlier we feed this butyrate, in agreement with what we're saying today, the earlier we expose animals to butyrate, the more effect we'll see. And that's clearly shown in even the regulation of milk composition. But some of the work we've also done directly in fat cells, again, trying to understand the mechanism, is to see what happens in the cell as far as differentiation is concerned. Again, when fat cells differentiate, they go from flat cells to round and lipid-filled cells. A lot of the genes that are involved in correct differentiation, appropriate differentiation, were induced in the presence of butyrate, especially people gamma, highly significantly induced again in these cells. But we were also interested in the expression of adiponectin. Adiponectin is a protein that stimulates or sensitizes to insulin. So insulin sensitivity, increased energy expenditure. With butyrate, we saw that there was increased adiponectin in those cells. So helping to promote insulin sensitivity, again, could be a mechanism by which butyrate could oppose type 2 diabetes, for instance. And again, it increased phosphorylation of AKT in these cells. So it did it in epithelial cells. We saw it did that in the brain, but clearly also did it in adipocyte. So activation of AKT, phosphorylation of AKT appears to be a common mechanism of butyrate in different tissue types. And of course, it leads to increased uptake of glucose, again, helping to prevent insulin resistance by increasing insulin sensitivity. Again, clearly helping to incorporate lipids into the cell, regulating genes that are involved in the lipid synthesis pathway. By also helping promoting healthy adipocyte differentiation. Okay, because what happens under certain circumstances, you have impaired adipocyte differentiation, and butyrate helps to stimulate that. And healthy cells, well-differentiated cells, are sensitive to insulin. But we also look at what butyrate does to release of lipids. Okay, to release of lipids, again, very complicated pathway, which I'm not going to go into too much detail, but we know that butyrate binds to these receptors, FFAR2, 3, and of course GPR109A. And this pathway is associated with regulation of release of lipids. One of the problems in obesity and diabetes is excessive release of lipids into circulation, which makes people dyslipidemic. They have too much lipid in circulation, and that lipid goes into liver. There was a question about fatty liver in chickens, but clearly butyrate can help promote oxidation or the storage of lipid in adipose tissue where it should belong, helping to, with more butyrate, you have reduced release of glycerol, a marker of lipolysis. You have that. But we think that effect might be coming through FFAR3 because butyrate led to increased expression of FFAR3 in fat cells, but downregulated FFAR2. So differential regulation of these receptors. So if we look at what butyrate does in adipose tissue, we know that it enhances insulin sensitivity, perhaps because it leads to increased expression of adiponectin, but that could actually be because it's a histone deacetylase inhibitor, which is associated with increased gene expression. So within butyrate could be leading to increased adiponectin via histone deacetylase inhibition, but eventually leads to suppression of lipolysis, increased glucose uptake, increased triglyceride synthesis, increased de novo synthesis, helping to enhance insulin sensitivity in adipocytes. So again, the level of butyrate that we see in circulation may not be as high as what you see in the hindgut, but I do believe it doesn't necessarily need to be that high. Okay, it can bind to its receptor, okay, and activate this gene expression that we're talking about. We just haven't studied or understood the system well enough to fully characterize it. And I think there's a lot more work that needs to be done in this area. Again, still talking about the PEG, we saw increased activation of this PGC1 in primary adipocytes, in muscle explants as well. Again, this is in agreement with increased oxidation, fatty acid oxidation in butyrate supplemented cells. And in this particular study where we fed piglets butyrate, again 0.3% butyrate orally given, okay, this by oral gavage, one by one pig by one by one, we saw increased body weight at day 84 in those piglets. Clearly, one of the things we've talked about in this conference is how soon or how early should you give butyrate. In this case, we gave them even after day 3 of birth. So after they have taken colostrum, we began to supplement them with butyrate orally, and we saw the effect in their body weight when we measured it at day 84. So clearly, very early on, you see some benefits. So to put everything in perspective, the things I've talked about today, we do see that butyrate coming from fiber or resistant starch or supplemented, okay, or supplemented can lead to, actually with resistant starch, can lead to increased microbial diversity. I have a lot of data on that which I'm not able to present today, but butyrate can help serve as energy source for muscle and help program adipose tissue for increased oxidation, but more importantly, it can stimulate protein synthesis, helping to repair that tight junction. Okay, helping to repair the tight junction, perhaps by increasing, also helping in adipose tissue, increasing adiponectin, and helping to enhance insulin sensitivity and glucose uptake. So it's produced in the gut, but it has effect in peripheral tissues as well. So butyrate acts, okay, on the gut. We know that, but it also connects the gut to the brain, okay, via the gut-brain axis. Either through the vagus nerve or even stimulation of neuropeptides and serotonin can directly regulate what is happening in the brain. Of course, I've shown data that also regulate peripheral tissues, muscle and adipose tissue, which is where I've done a lot of the work. But it definitely leads to promotion of integrity of the tight junction proteins, stimulates neuropeptides and hormones, stimulates metabolism, suppresses inflammation. I didn't show a lot of data on that today, but yes, we know that butyrate suppresses inflammation, perhaps acting as a histone deacetylase inhibitor and as a ligand for this GPR 41, 43, or also called FFAR2 and FFAR3 and GPR 109. So multiple mechanisms can explain butyrate effects. I just wanted to leave some of these questions or practical considerations for us. We've talked about some of them even during the workshop we had this afternoon. Question on age: when should we supplement butyrate? Is butyrate effect related to the maturity of the hindgut? These are things we need to really determine, because to really promote a consistent result of using butyrate, we need to understand its mechanism, but also when and how best and what level to use, especially related to the age and maturity of the hindgut. Level of stress, does stress level affect butyrate effect? We know what stress does to animals, disease, weaning stress. How about the level of fiber in the diet, the type of fiber, whether we use enzyme or not, xylanase and things like that. Probiotic use, do they interfere with butyrate effectiveness? Even antibiotic use, of course, you can affect the percentage or proportion of some of these microbes. But zinc leads to direct programming, direct shift of microbial community structure. How does that affect butyrate production? We don't know. And of course, several alternatives to antibiotics that are out there. We still have a lot to learn about how they affect butyrate production. Or even the environment, even the housing design. All these can affect local bacteria population, and all these can eventually affect butyrate that is being produced. What is the optimal concentration and form, coated versus uncoated, water soluble versus mixed in feed, pigs versus poultry? There's still a lot of questions that we need to answer, and I believe trying to understand some of the fundamental mechanism will be one way to go in trying to answer some of these questions I've raised here. So thank you so much for your attention.